Okay, please close the door, thanks. Please close the door, thanks. Please close the door, thanks. Okay, so we are now inviting Jaap Kaandorp for his lecture from the computational science group in Amsterdam University, a member of our academic board on this topic of model of embryogenesis and biomineralization. Thank you, Jaap, for being with us.
Thank you, Stefano, for your introduction.
I'm very happy to have you, Fred, and the other colleagues here. Thanks.
Thank you.
Enjoy your lecture.
Thank you. So, first of all, I would like to thank Stefano and Mauro for inviting me to this beautiful place for the opportunity to give a lecture over here. So, I will talk this morning about modeling embryogenesis and biomineralization. The organizers also asked me to come up with a little bit more nasty task. That's about the exam, which is needed apparently for this lecture. So I have it summarized on this slide. The idea is for the exam is to write it. You can write the essay about biomineralization in course, but you can also go to any other of your favorites, or that can be mollusks. You can also decide to go for forearms or coccolithophores. It doesn't matter, it's up to you. In the essay, I would like you to summarize what's known about this biomineralization process in this group. So then, especially attention to what's known about gene regulation and physiology. There may be a short discussion with what you could achieve with scientific visualization, with quantitative modeling, to get more insight in this whole process. And the essay should have a maximum of 4 pages and includes references. And you simply can send the essay to my email. So hopefully this is clear enough. So please let me know, interrupt me if there are any questions. Okay, if not, then on this slide, I would like to summarize the general interest of our group. And so we are very interested in the whole process, how the DNA is finally translated into some kind of growth form. And the general idea is that we have somewhere the genetic information, which is translated into gradients of morphogenes, which are controlling the processes in the organism. And for some reason, things are going. So these gradients of products, they lead to all kinds of actions. For example, they lead to migration of cells. They also lead to deposition of skeletal elements. And together with the environmental influences, they form a loop and make the final growth form. And we are very interested in this process. And the way we have been working to investigate this question is by using a combination of experimental observations, in which I collaborate with people doing experiments like Stefano or Frederick Marent. And in our group, we try to develop quantitative models of these processes to get some kind of mechanistic insight how these processes are working. Then the organisms, which I will use as a case, which we are using in as a case study in our group, are the so-called basal organisms. So they are. I don't see my.
You're very monkey.
I can point to the screen. The organisms in which we are interested are the so-called basal organisms at the bottom of the animal kingdom. And the organisms like the cnidarians and sponsors. And today I will focus entirely on the cnidarians. Okay. Thank you. So we are very interested in this group of animals. So, so one reason we are studying these animals is because they have all kinds of nice properties. They're also very suitable for computational modeling. And of course, for me, the first reason that I'm interested in these animals, because I think they're simply beautiful. That's my first motivation in looking at these animals. So I'm using two examples during the lectures. So we are using the CMMO nematostellar fragmences, which you see in the top, and we use the stony corals as another case. The nice thing, or the interesting thing of the comparison of both organisms is that the sea anemone is not calcifying, and the coral is calcifying. So, in a very initial naive idea which we had in the beginning, is that we can simply compare the genes expressed in nematostellar, compare them with gene expression patterns in, for example, Acropora, the coral, and find the genes controlling the calcification process. In practice, unfortunately, it's not that straightforward and that easy. That was the first idea. But by comparing a non-calcifying organism and a calcifying organism, we hope to understand more of this biomineralization process. Then the first problem is that it's a typical multi-scale problem. So in fact, we make a kind of a gap over astronomical scales, and you should be aware that molecules are described as length scales of angstroms, and so which is 10 to the power 10 minus 10 millimeters. Molecules also typically change on a time scale of nanoseconds. And so we are talking about seconds to the power minus 9. And we are interested in the growth forms which develop at the time scale of centimeters, meters, and which also develop in years. So, in fact, you have to bridge a gap in astronomical scales to understand this whole process. Furthermore, all kinds of different processes are interacting, which are working on different length scales and different time scales. So, for example, we start with the molecular physiology, that is defined on nanoseconds and angstroms. Then we go to the cell physiology, we go to the tissue physiology, and finally we go to the growth rate, which are all defined in very different lengths and time scales. So if you ever want to develop a model understanding biomineralization based on gene regulation, and so you have a major problem. So you have to solve what's known in computational science, the multi-scale problem. So you have to find some kind of trick that you can bridge the astronomical scales in time and space. So the kind of things which I would like to discuss is, I would like to start with models of gene regulation. I also would like to give a short introduction how to do this. Then I would like to show a case study. I would like to show the case study of nematostellar fragmences, how we can model gene regulation in such an organism. Then I would like to make the next step in the level of observation. I would like to go to the cells and the tissues. I would like to show a model, how we can model the whole gastrulation process of the sea anemone. Then I would like to discuss the physiology of the biomineralization process in somewhat more detail. And finally, I would like to enter the more microscopical growth models of the growth and form and the impact of the physical environment. So first of all, what can we learn? Gene regulation. So over here you see a picture about how gene regulation is, for example, controlling the body plan in a sea urchin and in the stomach. The usual way it's described in biological textbooks is that we make this kind of graph representations. In fact, the graph representation is pretty misleading because the gene regulation is also a process which is happening in space and time. So you can never only describe the representation. So what you see in the graph representation is that the graph indicates that some genes are connected with each other. So an arrow indicates an activation, and a block arrow indicates repression. And so basically, both organisms are using a kind of similar gene regulatory network to produce the animal. Still, they are different, and the reason why they are different is because in this gene regulatory network, a few things have been rewired, resulting in a complete different organism. And so, if you want to understand the growth and form of these organisms, you also have to understand how this mechanism is working in space and in time. Furthermore, so the thing which you cannot see in this regulatory network is the space, the spatial density. So if you make the sequences and you stain for gene expression in certain genes, so for example, you can find some differences between the sea urchin and the starfish. And certain genes, like the TBR gene, are expressed in very different locations in the sea urchin and in the starfish. So these pictures demonstrate that space can have a very important impact on gene regulation. Then a typical example, I think, of networks which you usually see in bioinformatics journals. So actually, this was a master project in our own group. We analyzed gene expressions which derives from data from microarray. And in this case, it was about neuroblastoma. The overall so already visualizing the interaction between the genes from these gene expression data was a very complicated task. And we ended with extremely complicated diagram. And if you are familiar with developmental biology, you may recognize some well-known developmental biology genes like Notch4. But the overall impression of the you get from these kind of diagrams is that they are extremely complicated. And in my opinion, they are also overcomplicated. And the reason for this is that you reconstruct the gene network, but you miss the temporal dimension, which is the genes are never expressed at a similar time, but they're also expressed in different locations. So if you miss the temporal dimension and the spatial dimension, you can never, from gene expression data, derive the correct network. And you get very overcomplicated networks. So my idea is if you can add the temporal dimension and the spatial dimension, you get much more insight how the gene regulatory network is working. So how can we do this? Now, so in modeling gene networks, you can include all kinds of beautiful information. In general, we want to keep things as simple as possible. What we will see is that even for very simple gene networks, we already quickly end up with pretty complicated constructions. So we have to think about this, and it also depends what we want to do with these kind of gene networks. So here is a very simplistic example of a gene network. Okay, so it includes a network of 4 genes. So we have gene A, which is self-activating. We have gene A is also activating gene C. Gene C is repressing D again. So, and we have a gene D. And gene D is also repressed by C again. And D is activating B again. And D is activating A again. So, the big problem is that we have a very simple gene network. But in reality, you have many more genes involved in the process, but they already can have a pretty complicated interaction pattern. So, how can we ever understand this network, how it develops, for example, in the temporal dimension? For the time being, I don't talk about spatial dimension, but how can we understand how it works in time? So first of all, we have to make a decision. So in this case, we know all kinds of details about the gene network. We also know that it, for example, is producing proteins, and proteins are binding to promoter sites, and they can work with the gene C. The helium AV works as a promoter for activating the gene C. We can include all these details if this is useful. You can also decide to make a simplification step and just try to consider to look at the gene network in this form, graph representation of 4 genes, but then add the temporal dimension. Now, how can you do this? For this purpose, all kinds of different approaches are available. So we can convert this to Boolean networks, in which the gene is on or off. This is an extremely simplification of the reality, and in practice, I think it does report very well. The first step which you can make is model these kind of gene expression patterns as ordinary differential equations. The next step is that we can add a spatial dimension to the ordinary differential equations and convert them in partial differential equations. And then basically you can model the whole process in space and in time. But doing this is still a major challenge. There's still people pay a lot of attention to this, but this problem of modeling gene regulation is still not very well solved. There are all kinds of complications. For example, a complication which you will see is that the gene regulatory network models contain a lot of parameters. You cannot find these parameters somewhere in the book, so you also have to come up with a method for inferring these parameters from real data. And of course, we also want to use these models in extension. So the next step is that we also want to couple these gene regulatory networks with other models, for example, mechanistic models of cells, and for example, biomineralization. So if you go to this example, the thing what we can do is we can construct, we can first make a simplification step. We forget about all the beautiful knowledge. We simplify the network of 4 genes like this graph representation. And then for every gene in this network, we construct an equation like this, an ordinary differential equation. And this means that in the case of A is only influenced by itself. And so we make a self-activating loop for A. And in the case of B is influenced. So I made a mistake. To be present. And in the case of B, it's influenced by itself, by C, and by D. And in the case of C, it's influenced by 3 other genes, and in the case of D, it's only influenced by C and by itself. So we need to construct equations for these 4 differential equations. And the way how to do this is by using this kind of equation. It's the Hill equation. By the way, you can derive these equations yourself, just by using the law of mass action. So just for my information, and for my curiosity, who of you is familiar with the law of mass action? No. It's maybe important. I think you have seen the law of mass action already several times. So, for example, if I have an equation something like this, you have an equilibrium reaction in which you have an atom A which binds to 2 atoms of B and forms a complex C. And then we can also say the complex A, B, C. And then you can mathematically represent this chemical reaction by the law of mass action. And in fact, you have seen it already a few times, I think. So, in which you take the concentration of your final compound, and you take the ratio of the concentrations in the reaction before the error. And so, for example, when we were discussing this kind of omega in biomineralization, you also see similar equations showing them, which are expressed in concentrations before the reaction and after the reaction. And this leads to this kind of law of mass action, but this is equal to some kind of binding constant. That is the ratio of concentrations before the reaction and after the reaction. And based on this law, you can derive all kinds of equations, like this equation. It takes a little bit of time, but then you can derive this equation. Basically, so everything, but most of the things we can see in computational biology, the equations are all going back to the law of mass action. So it's pretty essential. So in this equation, you see again a kind of binding constant. We have a kind of concentration of the substrate, which is raised to a certain power. And then with this equation, we can describe the reaction rate. No, so in a graph, it looks like this. So, for example, we have an amount of regulator in production rate. If we put the exponent to the value into 1, and then we get this signal curve, this red line. And if you go to higher exponents, we enter the black line, which is almost an on-off function. And you can try to represent all kinds of generating networks to give this kind of equation. And please keep in mind that it's all based on chemistry. And so in fact, we use a kind of law of mass action and to model interacting chemicals in the gene regulatory network. But then based on this formalism, then we can for these 4 genes, you can construct ordinary differential equations. For example, in In the case of A, the gene A is not influenced by other genes, and it's self-activating. And you can assume there's a constant rate of addition of the gene product to the equation. And the gene product A is also decaying in time, which is represented by this negative term. In case of inhibition, in which, for example, D is inhibited by C. And so if you have a larger concentration of C, and then the expression of D is going down, but also there's all the time a constant amount of D added to the system. And with this equation, you can represent inhibition. You can also include, you can also model the activation of two genes. For example, if the gene is being activated by A and by B, then if you have increasing concentrations of A and B, then the production of C is increasing. And again, there's a kind of decay curve. So every gene product decays in time. Think about the RNA, they are decaying constantly in time. And with this kind of system of modeling, then you can represent all kind of possible gene interactions. So hopefully this is clear. I don't want to bother you too much with the equations, but I also would like to demonstrate that from the things which I will show, how you can construct it yourself. And then the whole thing, what you can do is, as soon as you have these constructed these four ordinary differential equations, but for the four genes, we can model the time course of the four gene pairs in time. This is the time axis, and the y-axis is a kind of quantity. And you can now model how these things change in time. For example, one thing that you see, that's the continuous red line, is that there's finally constant production of A, and then the other gene products are fluctuating a little. It's a very simple system, it's only 4 genes. But please be aware that to model this system, we need the whole list of parameters, which is already pretty impressive. And over here, we just made a kind of guess about these parameters. In practice, we have to go back to the data and look at the data if you can infer the values of these parameters. So we end up with also a major parameter fitting forward. So, so there are all kind of pros and cons about this kind of approach. We can add a lot of detailed knowledge in these equations. The problem is you end up with many parameters which you have to estimate somewhere from the data. And in the differential equations, we don't have yet the spatial component. I will come back to this later on, how we can add this. And basically, it's, by the way, a very simple process. And if you have an ordinary differential equation for how x is changing in time, then you can add the diffusion term to this equation and add the diffusion equation, describing how the gene product X is dispersing over space. So it's not a big deal to make these equations spatially. Okay, then why do we want to do all these kind of things? I would like to summarize this on this slide. So our central idea in this case is that if we have a description of the gene network over here, and we know how these genes are interacting with each other, if we know how they can make spatial gradients over the population of cells, we can couple this with the population of cells And this population goes in combination with the model of the regulatory network can organize in some kind of interesting structure. So, so we can think about cells, and we can think about regulatory network. We can also think about a population of small robots, which can interact by local communication, and they can also make gradients over the population and organize in certain structures. So please note that this is actually, I think, a beautiful system, example of a complex system. In the cells, we only have local communication between the cell and the neighbor. So there's no central control in this whole system. So by local communication, and every cell has this kind of gene regulatory network. By local communication, we can get a very powerful complex system that is basically important in a kind of very complicated structure. And yeah, we would love to understand this kind of mechanism. So I made a kind of comparison over here between population of robots, but there's also an experiment which we did in the past. So, so then we also used this system of model of a gene regulatory network, which is used for local communication between the agents for making certain structures. So if you go to the new model, you see all kinds of different types of how these gene regulatory networks and cells are making configurations. So, for example, a very common way of development, which is shown over here, which is known as direct patterning. For example, if you know something about developmental biology of the Drosophila, early development in Drosophila, you get a pattern formed in the larvae, and so in this system, the pattern, the cells in fact are not moving anymore, but you get direct pattern formation, and that makes a kind of blueprint about all the things which are going to happen in fact in the future. In other organisms, you have in fact a more complicated mechanism. Then you have cells which can move around, and you have a regulatory network which in fact controls the movement and the interaction between the cells, which can move in the same structure. And the interesting thing is that probably in this case, you need a relatively simpler gene regulatory network obtaining the same result. But this, I think this is a very important part of the toolbox in the development of biology. Then just to show you for fun, this is the movement we made when we did this project also together with people working in the lab. So what you see over here is a simulation of the population of the moving robots. Every robot has the same regulatory network, just like a cell. And they make a kind of pattern where you get a blue gradient at the tips of the object. So, and the object is configured in such a way that it makes a body plan for a kind of starfish-like form. So again, we only use local communication. The funny thing is that people in science also published about this. They also produced in a starfish-like form with a collection of robots. In that case, they were just using a central computer, which were guiding the individual robots in the starfish field, which is working in a very different way, like biology is working. So the interesting thing in biology is that everything is working with local communication. And I just would like to show this in the movie. Now, only my movie is not working. Maybe it has to do with the cursor or something.
the video up here.
Go back to the arrow.
You can do it.
Yeah, it works. Thanks. So here you can see on how this this collection of simulated robots organizing in the starfish form. So the interesting thing is it's also a very robust shape. Every individual agent has this information to make this kind of starfish form. And you can even do experiments with this starfish form. And you can, for example, cut away an arm, and because of this regulatory network, the system can restore the original shape. So it can also reassemble it. So this, I think this also shows the powerful idea of these kind of regulatory networks. Okay, then what I would like to do is demonstrate how we try to do this with the sea anemone, Nematostella tectensis. And so over here you see a picture of Nematostella tectensis. It's a small sea anemone. It's something like this. It's a basal organism, so it's somewhere at the bottom of the kingdom. It's supposed to be one of the organisms which emerged very early on in the evolution. And we can compare it also with the borders, like Acropora. Important is with this organism is that the organism has been sequenced, so full sequence is available for the study. The kind of gene regulatory networks which we find in this are surprisingly similar in many cases in humans. It's in fact the gene regulation in this animal. The initial idea was it's a relatively simple animal. So the initial idea was that gene regulatory networks to make it, they're also very simple. One of the lessons which we learned by looking at this organism is that this is not true at all. And so the gene regulatory networks which are used in Amsterdam and maybe as complicated as we found in humans. So this was really a surprise for me. And I will show a few examples of this. Furthermore, there are several labs working now extensively in this animal. So there's a lot of information available about it. Then there are two technical things. So the area has so-called holoblastic cell cleavage, which is from a computational point of view very attractive. So if you ever looked at, for example, cell cleavage patterns in Shiona intestine as well, it shows an extremely complicated cleavage pattern, which is really beyond my understanding. I simply have no clue how it works. So fortunately, our pet organism is using a much simpler mechanism and is making an equal cell cleavage, which is not really anything special. It has also development by regional specification. So are you familiar with these kind of concepts of regional specification and versus motor development? And I would like to show it. Is anyone familiar with this kind of concept? Yeah, okay. Yeah. It's actually very It's actually very fundamental. So in regional specification, and so in fact the position of the cell determines the final fate when the cell is developing. And so you can also do transplantation experiment. And so you basically it's a very robust system. You get the same element. And you also have a sort of motor development. which is, by the way, found in many model organisms, also, for example, some rapids, and many insects, many, I think all mollusks have this kind of mechanism. So then, the cell fate is in fact predetermined. So if you are a certain cell, then it's already predetermined what you will become in the future. So, and so probably every cell already has a program in itself what it's going to do in later stages. So for me, it's still pretty ununderstandable how most of these development works. And it's so you need to understand the whole program in the individual cell, how it's determining cell fate, so what the cell will become in the future. The nice thing about animal cell is that it's not using this mechanism. By the way, also mice and humans are also using the same mechanism as animal cell, but they also have this kind of rapid regional specification, and they don't have this kind of very ununderstandable type of motor development in which everything is already determined what will become in the future. And in this case, we can also do something with our simulation models. That's the idea. One thing which is also an advantage of this is it has a relatively simple body plan. So later I will show that I can show the whole development of the organism actually on one slide. It never do this, for example, human. And so this is one advantage. There are all kinds of other strange properties in this animal which are very interesting. So basically, the animal is immortal. So if I culture it in the lab, if I take care of it doesn't get ill, it gets sufficient food, it basically will probably live forever. The other remarkable thing is that in this animal, never tumors have been observed. So it's probably a very highly robust mechanism of development which can easily repair mutations and problems. So so Nematostella is organic sequence, I think, which humans also would like to know. Although I think it would not be a good idea that humans would become immortal. Pretty nasty idea. So just to show, so how complicated the cell division can be in order to the cell division pattern in the tuning tent, which is by the way also used as a model organism. And it shows a very complicated cell division pattern, and I still don't understand how this ever works. Okay, then the relative simple body plan in another stellar advantage. So I can show the whole development of the organism from one slide. So we start with the fertilized egg. Then it divides, the egg divides in two cells, more cells. Finally, you have a bunch of cells which organize in a spherical structure. The spherical structure is hollow. And then you have a very important event at the picture H, where the hollow structure starts to invaginate in the gastrulation process. It's a very important step because it's the step in which the two basic tissue layers, the ectodermal tissue and the endodermal tissue, are made. So then the star indicates the future mouth of the organism. Then around the star, a few tentacles are being formed, and then basically the animal is ready. So at least the development process is relatively simple. Just to show you what's happening with this gastrulation. So you have the gene regulatory network, which is influencing the state of the cell. In the yellow part of the cells, the mechanical properties of the cells are changed. So the adhesion between the cells is decreased. And you get the information that these kind of yellow cells, which are known as bottle cells, they're almost becoming more fluid-like. And the bottle cells remain attached at the so-called apical ends, which are over here. And the whole population of cells is decreasing rapidly because of this mechanical event. The whole structure is being inward. And then finally, you end up with the organism, with the blue layer, which makes the active layer, the future skin, and also the nervous system, and the yellow tissue, which are the future intestines. Anyway. You might think it's just a minor detail from the development of biology. Why do we make so much fuss about this? But this is a famous quote by Rudolf Volcker, the famous development biologist. And he claims that gastrulation is a much more important event compared to other important events in life, because it defines the basic levels of tissue. Then, so the way we have been looking at images, we have been looking frequently at confocal light microscopy images. So the confocal light microscopy images were also made by the PhD student in my lab. He was originally trained as a computer science student, but he learned himself how to work in a confocal light microscope. So we have many pictures which he made in the past. So over here you see 3 examples of an animal which is gastrulating. Those are 3 different individuals. And in the right picture, we summarize the average geometry of these 3 animals. And red means in the form of the ectoderm, and blue the form of the endoderm. So over here, I summarize the shape of the embryo in time. So you see on top hours of the post-fertilization, and you see the shape changes of the embryo in time. And you see this whole embryogenesis event. And you can see that altogether this whole process takes about 14 hours post-fertilization. And you can easily observe this whole process. So you basically gastrulation takes place under a light microscope. If you have sufficient time, you can follow the whole process. Then we were interested in if we could reconstruct gene networks controlling these events. And then you have to think about the usual suspects, which are, for example, also relevant in humans. And the usual genes which are controlling the developmental process, like the Hox genes, the Green signaling pathway, and several other pathways. So we collected a lot of these gene expression patterns. So, for example, over here on top, you see an image of an Amsterdam hypertensus embryo at the point of gastrulation. So the star indicates the position of the tissue, right? We used a lot of in situ visualizations where it was stained for certain transcription factors or certain gene products. And so the blue circle is known as the so-called ONCOS1 gene. It's one of the Hox genes. And the interesting thing of this gene is that it plays a very fundamental role in specifying the body plan of the animal. It sets the so-called aporal axis in the animal. So at the aporal side, you have a huge expression of ONCOS1, and it sets up the first body axis in the system. So if we have the embryo, then this ONCOS1 makes the first body axis in the system, and it defines this axis in the system. It breaks the symmetry of the embryo. The question could be, what's initializing this symmetry break? The first symmetry break, what happens in the animal is the entry of the sperm that creates the beta-catenin gradient over the embryo, and actually opposite the beta-catenin gradient, you get the gradient of this antox 1. Now, we were very interested if we could quantify these patterns. So what you see over here is, so you go to the in situ utilization. So the in situ utilization is killing the animal. So you have all kinds of different individuals which have a slightly different shape. We go back to our average embryo geometries, and we adjust the average embryo geometry with this picture. Then we divide the image in small segments. And for example, if you start over here, in every segment, you can quantify how much blue stuff there is present. So you can convert the information in this image in a spatial plot that you see over here. So for example, if we start here, and we go in this direction, so you have a spatial axis, and this is an axis with quantity. You get a blue peak in the middle. And in this picture, you can include all kinds of other genetic information, other genes that are expressed in this study. And you still can use the radial symmetry of the animal. It only has, in this phase, one body axis. And you can show the whole gene expression pattern in a one-dimensional shown over here. But then, so how these genes are interacting with each other. So, already mentioned that the entry of the sperm makes the kind of maternal gradient in the system, which basically starts with the whole gene level. Then you get the information of the first body axis in the system on the one, which makes all axis. And then opposite of antox 1 expression, you get the expression of CA, which is a very important gene involved in the gastrulation process. It also affects the mechanical properties of the cells. And again, you can summarize everything in this picture. So we did this for all kinds of developmental biology genes. And so over here you see the whole list of 14 developmental biology genes. And the x-axis is a kind of time. So we made pictures of the gene expression patterns, spatial plots. The nice thing is that you can easily use these pictures as a kind of comparison in a kind of the cluster analysis of the whole data. We were actually amazed that our colleagues from developmental biology never tried to do something like this. Because it's a very nice way, I think, how you can organize this kind of gene expression data and can try to find patterns. And you can see that in the clustering process, you also get different clusters. You get a green cluster, you get a red cluster, chocolate cluster. And the funny thing is that these clusters also have a real biological meaning. And so we can divide them up in all kinds of clusters of genes which are responsible for the formation of a certain part in the animal. And so this analysis gives us a lot of information how these things are working and how these clusters of genes are. Yeah. Yeah. Yeah.
How many pieces for the analysis? Is that kind of error?
So basically, what you can do, the nice thing is that.
Sorry.
Yeah.
My understanding, you took the pictures of the gene expression and then
So first of all, we make this step. We converted the original picture into a spatial plot. We simplified the original picture. And we also, that's a kind of video which I didn't smooth these graphs a little bit so that you remove the stochastic process. And at that point, then you can very easily compare one picture with another picture. So there are all kinds of procedures how you can compare pictures in a cluster. Does this answer your question? Yeah, okay. Okay, then so the usual picture that you see in the textbooks is that we have bilateral elements and we have the Adrianus and sponsors. And some textbooks also claim that Adrianus have a radial symmetry, which is by the way not true at all. And I think if you go back to all the textbooks, it's already known that Adrianus and other Adrianus do have the bilateral symmetry. If you look carefully, we can see it, but they have a bilateral symmetry. So they are not at all radial symmetrical. And so the funny thing I think is that it uses a kind of similar system, which is also creating our dorsal ventral axis. That uses the total gradient of BPP, beta ventral teaching, which is coming back in non-BPP. But we make a second body axis. So we already have this body axis, and now we have the second body axis, which is set perpendicular to the axis, and then our radial symmetrical embryo changes into a bilateral symmetrical ordinate. And so what you see in the developing embryo, if you again look at the embryogenesis, you see certainly the asymmetric expression of hoarding, and finally, you get expression asymmetric expression of PPP and DNE in the order. And it sets up this second axis. So how does this work? So people encourage these kind of things. We see really so over here you see one of the genes which is involved in this process. Initially, you see it that it's symmetrically expressed over the radial symmetrical embryo. And then in a later time point, then you have to go to picture 9. You see similarly that it's only expressed asymmetrically. And then you have the formation of the second bond. And please note that it also happens at a very early stage, so already before the gestation event. And so this type of genetic a kind of system was proposed for this. So they proposed that there's a kind of network of genes involved in the gene interaction between PPP, quality, and the genes. But the problem in this paper was that they already assumed that there is an asymmetry in the so they didn't explain the asymmetry. The other problem is that, and I will show this in the next slides, is that what you see in the development of the interstellar embryo is that also wind signaling pathways are very important. And people are always looking at how these wind pathways are again connected in these kind of pathways. There must be a connection because they are both very important in steering the development of events. So about that was another surprising paper to me. So people also have been looking for wind pathways in another study. You would expect it's a very simple animal. So what is such a simple animal doing with so many wind pathways? I believe in total there are 12 wind pathways discovered. So apparently, for setting up a robust body plan, it needs a very complicated gene regulatory network. The way how wind pathways are expressed are also segmenting the embryo in different regions. And so the all axis, and the second is the electric axis in the system, then you can see that the wind pathways segmenting the embryo example, in oral part, in oral part. So, and that's a project which we are currently involved. So we would like to create a second gene regulatory network, which is also explaining this symmetry breaking, the formation of the second directed axis. And I would like to show a few examples of this. So the way we did this, we have used equations which are about similar to the Hubble equations, which I presented in the start of my story. There are all kinds of different alternatives, how to model the networks. It also demonstrates that the problem is not very well solved. And so in this case, we use the kind of approach by using these kind of Hill equations. And then the first regulatory network we modeled was the formation of the first body, where you have shown the expression patterns. The embryo is still radial symmetrical, so it can still show things in one-dimensional spatial pattern. And so we approximated this system with Hill equations, and then we have a kind of simulated gene expression over the one-dimensional spatial pattern. And I can only show it in a movie because it's a spatial temporal solution. So here you see the formation of the first body in the system. But together, at the science all kinds of origins which are involved in the controlling of the first events in the gestulation process. But in the final result is that we can model gene expression in space and in time. And so now we have a kind of representation of gene regulatory network, which is much more detailed compared to the usual graph representations that you see in textbooks. So we can, with this simulation model, we can demonstrate how these genes are changing in space over the internet and in time. So it can be different points in time in development. And you see, for example, the change of the scenario, back to this scenario, which is very important in the gestulation. By the way, the other genes, like the biomineralization, is also a very famous gene. It's coming back in all animals. It's, for example, also found in sponges. It's a very ancient gene. And it's also involved in the gestulation process. Okay, and then, so the second step which we try to understand if you can come up with a gene regulatory network which breaks the symmetry of the radial symmetrical animal as a kind of emergent phenomenon. And so we don't have to assume that there is already asymmetry in the animal, so we could explain it based on the gene regulatory. And this is our current proposal for this. So we assumed that there is an unknown gene W, which is connected to the other genes in this network. And the idea, the underlying idea, is that this W is also connected to the Wnt pathway. And so it should connect all the gene regulatory networks together. And we have, since we don't know this gene, we also don't know anything connecting. The way we solved it, we simply tried out all the possible connections. In this case, we can simply write them out, try them out all. And we try to find a network which is breaking this symmetry as a kind of emergent phenomenon. For this purpose, we use finite elements. We represented the embryo as a kind of face-like structure, tessellated triangles, And we computed this system of the gene expression mutations on this trace-like structure. And in certain networks, we could get a spontaneous symmetry in the design. And so over here, we see initially, so the embryos radial symmetric, and in time, this particular simulated gene shows an asymmetric system. And this is caused by the underlying mathematics which is used in the regular network. So we could get spontaneous symmetry gradient. And of course, the next question is, does it correspond to the real data, to the real observations? And for this, we again looked at many real gene expression patterns. Then again, when we go to our average shape of the embryo, and we adjust this superheat. You quantify the amount of gene expression in every segment in these animals. Now then, so if we still use the radial symmetry of the animal, and then we have again a spatial axis, which is this axis. We have again the quantity, and then what you see on these pictures are 4 important genes involved in the setting the axis of the system. So the measured gene expression patterns are these somewhat stochastic lines, and our simulated data are these continuous lines which you see in this graph. And if you are a little bit flexible, you can conclude that both the simulations and the observations correspond pretty well with each other. So over here you see biomineralization and mathematically, and then you can see that with the simulation, we can predict in simulation the gene expression pattern, which corresponds pretty well with the observed gene expression patterns. Currently, and I don't have results here. The interesting thing is that if you make exceptionally to this embryo, that it has an 8-fold symmetry. So it's 8 different segments. By the way, if you go to the stony corals, if you make a section in a line, it has a 6-folded symmetry. And so, which also explains the old-fashioned name hexacorollaria. And over here, you have the picture showing the gene regulatory network controlling this segment formation. And so, this is the next step which we want to make. And if you have the formation of the first body axis, the formation of the second body axis, and this segmentation ready, we almost can simulate the complete body plan formation of a developing sea animal. Okay, then I already mentioned that we are very interested in the comparison between the non-calcifying organisms and the calcifying organisms, and like the stony corals at the bottom. And so one of the examples which we also used is this particular the Great Barrier Reef. It's one of the first corals which has been sequenced, and in which also some developmental biology is available. So over here you see basically the coral line. So you see in picture H. So the coral, the planonia line is like this, and in picture H you make a section to the embryo. And if you look carefully at this picture, you see a 12-fold symmetry, and that is related to this 6-fold symmetry. And what you see is not a calcified skeleton here. And you see the expression of a gene, which in fact makes a pre-package on which place later on in the developing larvae this protein deposit. We are very interested in this process. Unfortunately, I don't have yet many details how this is working. The thing which we know about the calcified structure in the developing larvae is that it starts with this kind of, I can see on a section of the larvae, you see an eightfold symmetry. If you follow the development of the larvae, the 28 days, you see around this 12-fold symmetry, a very complicated structure with branching organelles being formed. And in TV, it's respected like this. So this also shows how we want to make the connection with calcification process. Now, if we look at the point, so at this moment, we have a model of the two body axes, the two body axes in the non-crystalline technology. We have a spatial temporal model available. We are currently also able to predict the outcome of perturbation experiments. And the beautiful thing about the development of biology of nematostellar is that so you don't use the usual mutations or something like in the formula, but that you can influence the gene expression patterns with morpholineage. So is everyone aware of morpholineage? Morpholineage, what kind of experiment this is? It's actually a very elegant and important experiment. So usually, so you have a certain RNA transcript with a certain sequence. For example, what you can do is you can make an antisense sequence, which is blocking this messenger RNA, which is called morpholineage. And you can block the expression very specifically of one gene. You can also even tune it quantitatively, adding more or less of this stuff. So I think it's a very beautiful trick. It's much more elegant, I think, than making mutations, because you never know the consequence of the mutation. And by the way, nematostellar is also very resistant to one-speed mutations. So it's extremely robust. But by my knowledge, there are no mutants known in nematostellar factors. Okay, then already mentioned actually the connection with the balls. For 12-fold symmetry. After that, the calcification stuff. And so then, of course, the big challenge in question in the future is to come up with the gene regulatory network controlling this calcification therapy. And we are very interested if we can handle this formation of 8-fold and 6-fold symmetries. Okay, maybe it's a good moment to have a break of 10 minutes. Gene, I know it's present in nematostellar. It must be possible. Yeah, so your question is if you can induce calcification in nematostellar.
Galaxies so important for classification, which first supposed to be non-calcified.
Yeah, so it seems to be involved in calcification, but it's also present in nematostellar.
The progress difference in morphology of geometry of the organism. The gene express. It's possible to identify the reason for whatever possible.
So there's a lot of tradition in development of biology, development process. environment and quality. So I know it's not a zebra fish that we use for developing zebra fish. You actually look into it and then you store the development process. I never heard these stories about him. No, I don't know this about Namaskar, but there are many other organisms with its deviations in the development process that we use to detect and pollution. My guess is that Namaskar it seems to be extremely healthy and robust organism. So it may be difficult, maybe it's possible. But by my knowledge, I have never seen deviating embryos of Namaskar. It's difficult to imagine. It's an interesting question. Yes, it definitely plays a role apoptosis to shape everything. But what you see in the tentacles is that it's actually connected to this formation of mesentery. So it sets up the eightfolded symmetry. And then first you get 4 tentacles. So this system also determines the position of the 4 tentacles. And then basically you get the expression of some tentacle formation involved genes, and then you need growth to make the tentacles. So at that point you don't really need apoptosis. But I can imagine that in other places apoptosis is also used to shape the embryo. It's an important mechanism. It makes our fingers, for example. Other questions? Okay, let's have a 10-minute break. Yeah, that would be great.
Was the microphone for the break? Okay.
Yeah, thank you. Okay, so a bit before the break, we discussed the modeling the gene regulatory networks. And the big question is, of course, of course there are cells. So the cells should be present in a model of an organism. And so, in fact, without the cells, we don't have the real representation of the organism. So again, I would like to use normal to stella effectensis as a kind of first example. One thing which I also forgot to mention before the break, I made a list of all kinds of properties which are very nice about nanostellar effectensis. And I think one property which I've still forgotten to mention is that the animal also has a high degree of regeneration capabilities. So you can take the sea anemone, you can bring it in a petri dish, you can dissociate the cells, and change the pollen into a population of loose cells. If you wait long enough, these cells are reorganizing into a complete polyp. So this animal has regenerative capabilities which are beyond imagination. But again, this brings us to the cells. And how do you model cells? Now, if you look in the literature, there's a long discussion about this. There are also many different types of models of cells around. This also describes that the problem is not very well solved. And so over here you see a few examples of models of cells which have been applied. Some people have been trying to represent the cells simply as spheres or ellipsoids. The thing that we would like to do in the model of the cell is using the so-called cell boundary model. So in the cell boundary model, we do worry about the micromorphology of the cell, and actually we need to describe the micromorphology of the cell. If you remember the slide that I was showing about the gastrulation process, at a certain point you get the expression of snail, which is influencing the mechanical properties of the cells. The cells are deforming and developing these kind of protein cells. So you need to have a kind of method available for describing the micromorphology of the cells. And for this purpose, we go to the cell boundary model. Again, I go to this to affect the developmental stage H, which the invagination process starts. And we start with the configuration in G. And we are very interested if we can explain how this process in a mechanical sense is working. How does it work? Structure in veterinary makes a two-layered structure. So again, we used the confocal light microscopy images, and over here you see a very good confocal light microscopy image of the sea anemone at the point of gastrulation. So in this population of cells, you see the population of cells where the cell adhesion is losing by the expression of snail. The cells remain attached at the apical ends. So they're still attached at the ends. Please note that the cells also have the polarity. So they always have an apical end and a basal end. In this spherical structure, the apical ends of the cells are organized systematically, and all the apical ends are at the outside of the sphere. So the cells are connected at the apical ends. The cell adhesion is decreasing. And the cells also make these kind of protrusions, the philopodia. And these philopodia are searching in the environment, and they try to separate with this rigid layer of cells, which will become the future ectoderm of the animal. And over here, you see several confocal light microscopy images of this whole process. Then the way how we try to model these processes. So, we used the method based on viscoelastic element. So we represent every cell as a model, and we subdivide the edge of a cell in sub-edges, and we load every edge with a viscoelastic element, which is an element containing springs. And we also load the simulated cytosol with a spring. So also the cytosol is now becoming viscoelastic. And so that means that if I have cells, if I press from there, you should conserve in this case area. And so if I press from there, it pops out somewhere else because of this viscoelastic interaction. And for solving this the spring equation, we use the method which we borrowed from molecular dynamics, which is known as the velocity per length numerical integration method. And so you can describe, just to bother you a little bit, the background of this. And so you can model the mechanics of the cells with a set of so-called Newtonian equations, and spring equations, and like this. And we use a kind of method for conserving the area in the cell. And you can use this kind of equations for this. And we can also represent contact forces between cells, again by using springs. And we can also simulate the ferropodia as a kind of mechanism for long-range adhesive mechanism. So you can use the ferropodia, explore the environment, and it finds, for example, new locations for the cell. Now, why were we so interested in doing this? So, if you look at the confocal light microscopy images, you see that there are many individual processes going on. So you have lowering the cell adhesion, you have the action of the ferropodia, and how do they interact, and how do you finally lead to a kind of distillation process. The other remarkable thing is, I think, if you look yourself at this kind of distillation process in your microscope, the whole process looks pretty messy. But the remarkable thing is that it always ends up in perfectly distillated envelope. So it's a highly robust process. And how does it ever work? So that brings me to the next question. So, although we observe a large variability, we always see perfect distillated embryos. And how does it work? Now, again, the representation of the cell. And so the cell has the cell is a protein. That's an apical indication. And the cell is divided into sub-sections, and it's represented as a polygon. And for example, if you bring two simulated cells next to each other, you find the cell that between two neighboring cells, then you get this kind of solutions for the system. So you can try to find some kind of minimum energy curve. Then the initial point of the simulation, but it's a spherical object, just like I have shown in previous pictures. It's of course a two-dimensional representation. Due to gene regulation, you have a population of red cells, and in reality, this is gene regulatory network. It is expression in this population of cells. And in the red population of cells, we now going to lower the cell activity. And this is actually happening over here. When we lower the cell activity, the structure is bending in. But you don't get, if you look at the picture, you don't get a fully castellated embryo. Then simulated with a polygon. So over here you have a cell which is attached over here at the rigid surface. Here you have another rigid surface. And now the cell is going to make in a stochastic way philopodia, which explore the environment, and they try to zipper with the environment. And now you see a cell which is desperately trying to zipper the rigid surface. And now we bring everything together. So we have again population of cells in which snail is expressed. Going to lower the cell adhesion. We're going to make the philopodia, which try to zipper with a rigid surface, green layer, and we can do the simulation of the gastrulation process in 2D. You see that we get formation of bubble cells. So the nice thing is that the shape of these cells corresponds nicely to what we see in the bubble method. By the way, they also use this movement nowadays to explain students in development in biology how gastrulation is working. And you see that we enter in this case with the regulator in the blue level. By the way, please be aware. So what I'm showing here, it's a very short movie. That originally was done on a PC, and it took about 6 hours of computing to get this movement. We did all kinds of clever tricks that we can do it using GPU computing, by which we can do it almost in real time, so, which was an important step for us. I then already mentioned that we are interested in the diversity of the model. So over here in this diagram, we have different model parameters at, for example, an error model parameters which we vary in parameter variation study. So what you can see is that for a broad range of parameters, you get more or less reasonable gastrulated embryos. Only in a few settings you get embryos which are not perfectly gastrulated. So this shows that the model is fairly robust. It works over a long range of parameters. You could argue you get heterogeneous embryos like this over here, but the interesting thing is that in this case, this kind of types of gastrulation are also found in, for example, the sea urchin makes this kind of embryos, and it's using this open space for forming the first skeleton and for the insert of the biomineralization. So the thing is that part of this parameter space is maybe used by other embryos. Now then, the remarkable thing about gastrulation. So if you go to books about embryology, and so for example, if you look at biomineralization, the book is actually in Russian. It's a beautiful book describing the comparative embryology of many different opinions. It's really a pity it's in European. But she shows this picture for different types of embryo, which have different mechanisms to make this gastrulated embryo have 2 layers of tissue. So you have all kinds of, if you look in metazoans, you have all kinds of different pathways to make this gastrulated structure. So the pathway which I discussed now is the invagination process. So the cells remain attached at the apical ends. So one question which you could ask immediately, what happens if I release this condition? For example, if the cells are not attached at the apical ends. This is, for example, happening in this pathway, where the cells are simply released and moving towards the inside of the body, and you get the same result. And so you have different mechanisms to make the gastrulated embryo. And it's actually very strange, it's a very fundamental process, that there is so much variation with this process. Yeah? So, sorry? This? No, yeah, so that would require a much more detailed cell representation. So then you would need to represent so the entire cytoskeleton in the cell. It's possible. People have been working on these models. The price that you pay is that you get a very complicated model. And in our case, you already have a pretty detailed model. So that's good. People working on cell models, they use much more higher level of abstraction models than we are using. In a model in which you also represent the cell polia, so again, you're really into troubles. And we didn't need this kind of complicatedness in our models. Explain the things which you are looking later. If you could come across this kind of problems, of course, you have to reconsider this. Maybe also add some representation of the cytoskeleton. Yeah. Then, yeah, so I mentioned this, so there's an alternative pathway to get castellated structures. The strange thing is that it's, for example, found in other Nigerians. We want to know where going. And this shows in the picture how this castellation works. So when the cells at the bottom of the embryo, they are changing the mechanical property, and they are popping each other, and then finally make the inner layer of endodermal tissue. So this is an alternative pathway to castellation. Now, we were very interested if we could do the same thing with the same model, but by just the only modification which we now make is that the initial condition is a little bit larger because also Biglia starts with a little bit larger spherical object. And now we effectively forget about the whole condition that the cells are attached at the apical ends, so the cells are freely allowed to freely move around. And then, so we get this kind of vision, that we get the model where the cells are ingressing, and then we to red layer inside the blue layer, and you have the same result here, again, castellated embryo. And so with this model, we could even confirm some kind of experiments with normal morphology. Okay, the problem is here, at the bottom, you see the real observations. And again, this is a parameter variation study. Okay, then the next question is, can you explain everything in the three-dimensional manner? Now, so this is of course not true, because already mentioned that the animal has a bilateral symmetry. So finally, if we want to go to a full polar, we need also a two-dimensional model. And in fact, I will also show a case in which you really need maybe also for the castellation process a 3D model. So it's a effective project in which we have been working for a long time. I almost don't dare to say how long we've been working on this. It's almost, I think, for a decade. But quite soon we are going to publish this. This will be a cell-based model which is working in 3 dimensions. And just to show you a few aspects of this. I first would like you to show you why we ever need such a model. So if you make a scanning electron microscope, then you get this kind of pictures. And if you look, so you can make a section to the embryo, then you see the castellation process. But you can also look at the future oral side embryo. And then you see, you can distinguish, if you are experienced developmental biology, you can distinguish the pre-embryonal plants, so the population of cells, which will become the future population of embryo. And you can see it actually from the shape of the cells. So the cells in the pre-embryonal plate are a little bit more rounded, and they are in this environment. But the strange thing is, already mentioned, that looking at the developmental process, things really look very messy, and very irregular. And also the shape of the pre-endodermal plate. And if you make it wrong with it, the bone really apart, it has a very irregular shape. And also, if you compare one individual with another individual, you can come across all kinds of different shapes. Of course, if you think how the whole system is working, it makes sense. So you do this gene regulation with chemical agents. And so the chemical agents diffuse a little bit over the cells. You have all kinds of stochastic elements. So also small amounts of this chemical stuff are being produced and are dispersing over the cells. So it makes sense. It's very irregular, but what's quite difficult to understand. How is it possible that this always works perfectly? So you are very interested if you could develop a three-dimensional version of the gastrulation fish. You need to test out if the model still works for all kinds of different shaped pre-endodermal plates. And so the cells, the representation of the cells which we are going to need are actually derived from this icosahedron. And in the icosahedron, so every vertex is located on the surface of the sphere. What you can do is you can subdivide every triangle again in 4 triangles and project the new vertices again on this sphere. And starting with this icosahedron, you get easily a triangulated sphere. It is every time closely approximating the real sphere. And we use this kind of representations. It's a kind of initial representation of the cell. And so please be aware that in this three-dimensional model, we are going to define viscoelastic element over the edge of this triangle sphere. And so we have to complete now the forces in 3D on all these edges. So the computational effort will be dramatically more. So here you see the first experiment with a group of triangulated spheres with those viscoelastic elements. We pack the spheres on top of each other, we compute the equations, and so the cells again assume a kind of minimal energy configuration. And the nice thing is that we immediately get the right shape of the cells. And so in the two-dimensional model, I assume that we start with this kind of configuration, that the cells are more or less cone-shaped. I don't have to make this assumption anymore in the three-dimensional model. And so I automatically get the right shape as you mentioned phenomenon. And this is usually something which makes it happen. The price, of course, which we have to pay is a lot of more computation time, but we can drop one assumption in the model, which is always important. And so here you see the three-dimensional cell. So please note that in reality, we 4 kinds of these cells. We are currently working in movies with 4 kinds of cells, which will take an enormous computation. And that we can get simulated gastrulation structure also in our three-dimensional model. So you now see how the future is bending inward. And then back to the original question. So can our three-dimensional model stand all these kind of irregular shaped pre-endodermal plates? Now, we really see the set of experiments. We experiment with all kinds of shapes of the pre-endodermal plate. And you can see that at least in the fixed pictures, you still get reasonable gastrulated structures. But only a very weird initial configuration of the system fails. This also shows that with the three-dimensional model, you still can deal with these irregular shapes. Okay, then so we need these cell-based models to understand morphogenesis. The two-dimensional models, which we have, are very robust. We can explain all kinds of details of the gastrulation process, and like this apical constriction and the body cell formation. We have the first version now of the 3D model, and one step in which we by currently is we try to make a direct coupling between the gene regulatory networks and this kind of cell-based model, and also of course with biomineralization. Okay, this ends this set of slides. Are there any questions? So I forgot to ask you, please interrupt me if you want to know something. Yep.
What are the limits of insurance? Or is be able to now is something really is it because the cells are still or because
So one problem is it will be very hard to find the reality the gastrulated embryo in which something is going wrong. I have never seen such an embryo. And so and please also note that this is part of the story. And so my idea is also that, and so there's a good reason that these gene regulatory networks in CNM are so complicated. And so except for all these mechanisms which are presented over here, and there are all kinds of other mechanisms in the gene regulatory network which take care of this gastrulation is working perfectly. But for example, also some genes, especially expressed at around the mouth of the embryo, they also influence the stiffness, so they can also deal with this problem. But of course, if you want to investigate this, again, you need to go to the 3D model. So, yeah. This explains, yeah. Oh, wait, I know what I have to do. Yeah. Yeah, okay. So this is the second part of my story. Now what I would like to do is so we talk about deregulation, we talk about cell-based modeling. I briefly talk a little bit of things about why we need to. And now I would like to go to one of our, I think, active points of research. It's trying to model the classification physiology in corals. And after that, we again increase the level of observation. And I also would like to go to show you if you're more macroscopical modeling of classification. And if I have sufficient time, I also would like to show a few things about how you can quantify and analyze complex shaped three-dimensional forms. So again, we were very interested in comparison. And again, please keep in mind that we are dealing with a multi-scale problem. And the kind of level of observation where I would like to go now are the things which are happening at this level of the observation. In fact, at the tissue level, so we are going to try to construct a modeled tissue physiology. And I'm very happy that both Frederick and Stefano already introduced a lot of these kind of things. So, as an introduction, still some pictures of the Great Barrier Reef in Australia. And then we are going to look at the ball. At the picture left, you see the clean skeleton. And at the picture right, you see the polyps covering the skeleton. And this was already mentioned a few times, but this will be again important also in this story. And so the polyp is in fact sitting on top of the skeleton. And in the cells of the polyp, you have the zooxanthellae, which are taking care of the photosynthetic process and providing energy. Then the very important problem, especially in the Great is this coral bleaching. So if the temperatures are getting too hot, the coral is stressed. You can have a condition that the coral gets rid of its signals and the coral is bleached. And if this takes too long, the coral won't survive and it will be bleeding. And so the nitrogen coral, the coral enter the bleached coral, which will die. And now I don't have to show this picture again, but always I think pretty sad. Then the summarized the main chemical equations in the life of the coral. You have the photosynthetic process, which is basically absorbing CO2 and making sugar. You have the respiration, which is using the sugar to produce CO2. You have the calcification, which is in fact also absorbing the CO2 again. Producing the calcium carbonate, and we have an important reaction. And of course, people are very impacted. What happens if this whole process is acidified, like Stefano yesterday explains? Does it still work? And this kind of picture from yesterday, this shows CO2 concentration in the atmosphere over time. And what you can see is that you currently experience a period in which the CO2 level is at least for recent time, increasing values. So what are the consequences of this calcification of physiology? Now then, how does the whole process of biomineralization works? So this is actually a picture of a very famous book by Darcy Wentworth Thompson, Growth and Form. If you ever have the possibility to read the book, I definitely advise you. It's a beautiful book. It contains many examples. It contains a chapter with examples about it. It calls it conclusions in speakers. It's about biomineralization. And it shows all these kind of beautiful shapes that you can find in nature. And I think the frustrating thing, I think until now, I think that no one fully understands how these structures, how these beautiful structures emerge in biology. No one understands, let's say, how a living organism is creating beautiful things. And this will be extremely important in fundamental knowledge. Okay, then the idea which we are using in our knowledge of biomineralization. So we think that there are two secrets in the biomineralization. You need to kind of confined space. And this is a very narrow space. The confined space can be like in a coral. So you can have the coral tissue and the cells sitting on top of the coral tissue. And this confined space can be cells in the skeleton. But It can be also, for example, in the organism in itself, in the unicellular organism itself. You need some kind of gene regulatory network to control this transcription, but that the right things are happening at the time. You have all kind of channels and pumps which are controlling the concentrations in the confined space. And so I would like to emphasize that it's very important in the confined space that you have the right chemical conditions for getting the deposition of biominerals. And then the other secret, I think, of biomineralization is the organic matrix, the proteins which are interacting with this crystallization process. And again, there's very little known about how this interaction works. But finally, this leads to the formation of all kind of beautifully shaped crystals. So this is our idea how we should understand in a model the biomineralization process. But then we start with the work which we did also together with Frederick. And so then the first question in this work was, what kind of proteins are around in the body? So for this research, we investigated agropolar and nelopolar skeletons. The skeletons were cleaned and they were grounded and finally dissolved. And the final result is that we used the calcium carbonate and the two components between the soluble matrix and the insoluble matrix. And based on based on the step, so we use these proteins first kind of peptide digestion, and we simplify them in peptides. The peptide finally back. And finally, the protein identification. The result of this work was that we could get sequences of the proteins involved in this calcification process. So we ended up with a surprising long list of proteins, and also by it also involves some kind of bioinformatics work. And the final result is that we found a large collection of proteins which are involved in this calcification process. Now then, we did find all the proteins for which we know some kind of function. Some of the proteins have enzymes, some of them are acidic proteins, and we could make a kind of overview. And so the remarkable thing is that, first of all, I was really surprised how many proteins we are finally found in this method. And so in orange here, you see the proteins which are characteristic of which were only known in protein. So you can see that there were also 3 proteins that shared which are present in this way. And this brings me immediately to Stefan's question about the galaxy. So you can see that the galaxy is also present in non-interstellar. And for some reason, this galaxy is also seems to be involved in the calcification. And you can also find the proteins involved found in iron. And so we ended up with a very strange list of proteins. And one of the most remarkable experiments, which Paula did this, Paula Silva, who did this work, presented finally in my opinion, this picture. I was really amazed by it. So over here, so if you did a kind of a experiment, you see both this kind of soluble matrix in a calcium carbonate. So On top left, you see the blank experiment. And so then you get the regular calcite question. And if you increase the concentration of this cylinder matrix in the crystallization pattern, you can see that you are strongly influencing the crystallization pattern. You get very strong crystals, but you don't get calls. So now, in fact, we only have an additional problem. You can see that it influences the crystallization pattern. We still don't know how it ever produces a call. And I think for this reason, we have to go back to this confined space, to this whole idea about how things are controlled. Because these kind of proteins are released at certain places, at certain times. This is controlled by gene regulation. And you also need the kind of mechanisms that you get the right concentrations in the confined matrix. Now that brings me to the next project. So this is the project in which we are still working. And this is the first presentation about this work. So in this project, we try to model the physiological conditions in a confined space. So what we are going to do is we are going to go. Please remember here is the global scale problem, and we are now at the tissue level. We are going to model this small part extracellular calcifying medium. So it's located between tissue and skeleton. So there's also a lot of literature present in which it's stated that photosynthesis is influencing the calcification process, the one of these calcifications. So the other big surprise is, if you look what's published about this, is the enormous amount of hypotheses. The other frustrating thing is that some of these hypotheses are also contradictory. And so one of the ideas was, if we make the quantitative model of this whole process, then maybe it's possible to get rid of some of the hypotheses and find the hypothesis which seem to correspond the most with the real observations. For example, so everything is, by the way, very nicely reviewed by paper. There are many different hypotheses on how, for example, calcium is being transported over the cells. And also about the light of this calcification. There are many different hypotheses available. So we would like to find a way in this pretty confusing world of different hypotheses by developing a spatial temporal model. So the aim was to develop a kind of model which works again both in time and space. There's a very good reason to go to spatial temporal models. Currently, there are all kinds of developments. For example, it's a part of the newspaper that people are working with, for example, pH-sensitive dyes, in which they can visualize in the tissue the pH. There are also techniques that you can visualize the concentration of calcium ions in tissue. So you have all kinds of beautiful pictures and beautiful spatial temporal data available. And at this point in time, I think it's also very important to come up with a spatial temporal model, trying if you can quantify and explain it. And of course, you would like to use this model to test out and maybe create even new hypotheses. But then the model which we use is, of course, as simple as possible. But that's always, I think, our guideline. And you should not introduce complications if it's not needed. So use a very simplistic. So we have a picture number, which in the contains the photosynthetic result from camera. There's a surrounding. Sorry, there's the extra cellular classified medium, and there's this skeleton. And these measurements, as you see, are taken from the electric. We model the whole system with the system of coupled reaction between this includes the entire chemistry. And in this case, only CO2 is allowed to diffuse over the cell membrane. It's a relatively very small molecule. And then, so some kind of representation of the calcification model. So we have the C model, which is the source of the calcium gas. We have the C model, the source of the bicarbonate and the inorganic carbon. Part of this is used for the photosynthetic process. We have a transporter, which second also mentioned yesterday, which is an extension of sequence for T12, which enters in the surrounding. And we have the respiration, which is adding CO2 into the process. A very important step is this enzyme. Without this enzyme, nothing works. That's the carbonic anhydrases. And you stimulate the reaction. And then further on pH, the carbon molecule concentrations, and we get the calcification. And of course, we now represent it as a simple deposition reaction, but we already know that in reality it's much more complicated. Although that will be the next phase in the starting model. The important thing now is that in the college that when you function role and the details and light becoming further. It's also known that the calcium activity is switched on during light periods. And the carbon anhydrases is very important.
Another question.
Yeah.
How do we connect the photosynthesis? Because the patient will be over.
I will show you after the steps. Yeah.
Okay, it's more than your guys' investigation.
Yeah. So in fact, you have competing processes. So photosynthesis wants to use CO2 for making sugars, and we also need CO2 for making calcium carbonate.
Maybe a land, I pronounce it.
How can you? How?
The land.
Yeah. So the idea is and so
The land, because it's land.
So to betray already a little bit the plot is the story, so the photosynthesis. So this protein, the calcium reality, so every step will cost 1 ATP molecule. So it's a pretty expensive process. So you need energy to do this. And you need quite a lot of energy to get the correct pH. Sometimes you also need fluids in the literature. For example, it should be possible that calcium can freely move between the cells. So if you need a very high pH, so you already can see from the quantities that this can never be true, because it takes a lot of energy to pump away sufficient proteins to get the correct pH calcium deposition. So the dotted line shows one of the hypotheses which is around which we think can never be true. So the photosynthesis is producing the energy. It's also contributing to by also contributing to the correct pH, it's also increasing the pH. That also helps for the calcification. But to explain this, we give more detailed model as a part of synthetic growth. That's one of our future aims to explain that. Then, so in the spatial model, we try to evolve the most relevant chemical qualitative, controlling the chemical composition of the ECM. So there's also diffusion included in the models. So we use coupled reaction diffusion models. And so we also have been looking around in the literature. So how many people have ever followed the pH, for example, in this extracellular calcifying medium in time. And so we need a time series because we know the pH to light and dark conditions, and preferably over a range of time. You need to know the pH, for example, in ETM. And we also would like to know it in the other compartments of the model. Of course, it would be also interesting to know the calcium concentration in the different layers. And so, to check if we can predict these kind of values with the model. Now, we have been looking a long time around in the literature, and actually, there's very little data around about this. One of the few players which gives a kind of complete time series about this, if you take it in Adrianus in 2003, he actually measured these concentrations in a very crude way, just used probes, entering these very thin layers of tissue. So some people also have been wondering if this is really possible, but he didn't. And he made these kind of time series. And by my knowledge, this is still the only really temporal information available about it. So we were interested if we could recreate the data in the simulation model, which is validate if the outcome of the correct. Can we also get with the model some more understanding? So how the coral is keeping the right chemical composition in the ECM, and can we use this model to reproduce the light-dark dynamics? That's the effect of light-enhanced calcification. And of course, another question that we mentioned here is, can we predict what's the impact of acidifying seawater in the whole process? Now, in the whole modeling process, we use a CO2 chemistry system, which is described in the paper by Jacobus. And we use a very simple topology. The dimensions of the topology correspond to the tissue layer. And so we have in fact now 5 components in the model. You have the seawater, you have the cell lampion, you have the cells covering the ECM. So the kind of lost cell, and you have the ECM itself, and you have the scale. Now then, how do we represent? So, for example, the calcium Adrianus activity. We then use mathematical equations based on this law of nitrogen. If we model the flux of the mechanical reaction stress. You can also model the activity of enzymes with this kind of equations. So it can be also model carbonic anhydrous with a switching kind of approach. And so over here you see the effect of the diagrammatic overview of the model, based on the magnetic material. And first of all, we have a flux of calcium ions for the calcium anticonvulsant, which equals the calcification rate. And the CO2, the CO2 is into calcification. And we also looked at the system, reaches a steady state, close reach a steady state. And so we still make a few assumptions. So we think there's one stable equilibrium. We can also actually demonstrate this in the simulation model. And so the other things, what we try to do is that the chemical concentrations corresponding to this equilibrium point are controlled by the chemicoblastic cells. And by understanding how this stable point is controlled, we try to simulate the light-darkness dynamics as observed by all around. Now here you see how we investigate the steady state. You can start with all kinds of points with total autolimity of the picture. Then you can see environment. You can also start with all kinds of initial conditions for the inorganic environment. And you can see in this kind of phase-like diagram that the trajectories always move to a kind of stable state. And so that's the way how you can mathematically analyze this kind of system. And then, so the final comparison. So our model results are shown in solid red lines. And we have the experimental data, which is shown in red area. And so what you can see is that actually, maybe you don't believe it, we are very happy with this field, because at least qualitatively, so our simulation model comes pretty close to the red observations, the original observations, over the long. So this means that in time, you can reproduce the pH in the model, you can reproduce the calcium concentration, and you can reproduce the saturation step. Very important. We also tested out if the model is robust. So I think one of the secrets in biology is that every part of biological process shows a high degree of robustness. Also, this model shows a high degree of robustness. So this is also a very important aspect of the model. Then back to the hypothesis that we have tested with our classification model. So there are a few papers around we claim that only paracellular transfer, that only paracellular transfer is sufficient for classification. This can never work. You can complete it. So we also tested out a few hypotheses how light-induced calcification is influencing the calcification process. And in this case, it has a kind of limited effect. And so calcium alkalinity controls the pH in each limb. But it still has a kind of limited effect in the calcification. And so we know that every step requires ATP, so we also need the energy to do this step. And so there's still an influx of CO2, which is also a rate determining step in the process. The other thing that we found in the model is that the process only works with calcium carbonic anhydrase, that really essentially getting the calcification going. So this is a very important step in the process. So based you could show that the effect of paracellular transport is linked. The only pilots transport is never sufficient. That can supply range to the ECM, the dosification. The carbonic anhydrase is essential within the dosification. The light enhanced dosification is involved two processes. So we have more available respiration of CO2 and increased activity of the calcium and the acid. So the respiration is another important conclusion, which I didn't list over here, is so if you go around in the literature, so there are many, I think I have listed 14 different hypotheses. But one of the hypotheses is that you have the cardiovascular cells and turning of the skeleton, and seawater can freely flow out and in between the cells. So I believe you can see from these kind of quantitative conclusions that this can never be true. And so if you would allow these kind of openings, then you need immediately lose your expensive gradient, your expensive high pH value, which you need to do classification process. But because opening these pores, you immediately destroy these kind of chemical gradient. And so we think you can verify and you can actually reject the hypothesis by using this kind of model. Okay. Any questions about this problem? Yeah. 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 So. So I think the story about vesicles which are transported from one side to the other side, I don't buy it. I think there are no green men in the cells who are helping this process. No. So it makes things much more complicated. And you should explain this vesicle information. How is this ever controlled? It's difficult to imagine. Yeah. That looks to me the only possibility to get the right concentrations in the machine. I think. You need to get rid of all these proteins, and you need to get the right pH. Okay, then I would like to continue with but maybe it's good to have a break now. We're talking for about an hour.
1 hour.
Yeah, let's have a break of 10 minutes. And please interrupt me if you have any questions. Sorry? Yeah, the mic. Yeah.
Just for
Thanks. Okay, so what I would like to do now is go to a yet higher level of organization. So during this presentation, I'd like to focus on microscopical models of growth and form. So before I start introducing these models, I still would like to introduce a big issue in corals, which is the influence of the environment on the growth process. And just to try to convince you that there's an important relation between the growth of a coral and the impact of the environment. So over here you see the technology in the coral from the Caribbean area. Over here you see shapes of the coral collected different depths. Both at shallow locations, it makes a spherical form. And if you go to deeper locations, shape of the coral changes, and it makes more flat surfaces. And so you can see that there's a strong connection between growth form and environment. Over here you see a coral which we have used many times in our research. It's another feature from the Caribbean. It's not runs in the area. And you see that it develops on shallow locations, more compact density thing. And if you go to deeper locations, it makes branches which are more open. And the example of this is taken in the big bang theory. So that's closely following big bang theory. That's the left top you see the typical growth from the sheltered location. It makes very thin density. And if you go to highly exposed level, it makes very dense impact areas. So hopefully with these pictures I can convince you that there's an important relation between the growth process and the environment. Also between flow, because it's important impact on the growth process. So over here you see the reconstruction of the coral acropoline decentifier, collected by the coral institute. And this is a typical low flow morphology. And if you go to high flow conditions, the shape of the colony entirely changes and it makes a much more compact shell. This has also important consequences. This also has to do with the calcification physiology and the photosynthetic process. And the photosynthetic process is producing energy for the calcification process, but as a byproduct, the photosynthetic process is producing liquid oxygen. For the coral, oxygen is poisonous. It's an oxidative agent, and it interferes with all kinds of biochemical processes. So effectively, oxygen is a waste product. And if you imagine that we are a coral, and the photosynthetic process is in a high degree, and then, for example, between the branches, you will get relatively high concentrations of oxygen. And the oxygen interferes, for example, with the photosynthetic apparatus. It effect can destroy it. And finally, it can lead to bleaching. So what you can see is that the water movement has an important influence in this process. And so it's a waste product. It's, for example, accumulating between the branches. But if you have sufficient flow around the branches, then the oxygen can be effectively flushed away. And you can also see this from papers in which they're computed, in which they calculated the survival rate of corals for certain flow velocities. if the velocity, if the ambient flow velocity is high enough, then the coral can survive. But especially in sheltered locations. So due to this issue, the corals are a big problem. Then there's another issue in which we have worked. So that has also to do with the symmetry. So maybe you notice that we are fascinated by symmetries. So what you see in many branching corals, which you can also see in the previous pictures, is that these corals more or less have a kind of, at least in a statistical sense, like a radial symmetry. And so branching corals seem to be flowing in all directions. So you also know that most of these organisms are living in marine environment, and basically all these organisms are at least twice a day exposed to the tidal movement, in which the flow direction is reversing twice a day. So it's a very interesting paper of colleagues Israel, and they did an experiment in which they did a personal coral colony in a unidirectional flow. And by using the flow, they exposed the coral for 1 year in directional flow. And they were interested in influencing the symmetry. And you do see important effort. So you can actually see it on these pictures. So over here you see a kind of metal screw, which the coral fixed into the plastic. The arrow indicates the direction of the direction of flow. And you can see that the morphology and symmetry of the colony is changing. The branches are growing towards the flow. And another important observation was, if you do this experiment in a relatively low flow conditions, then especially at the the upstream side of the colony, you get a problem with the accumulation of oxygen, but also local heat. And also you see the whole experiment in which the colony is simply exposed to the bidirectional flow in regular symmetrical shapes. So again, you see that influencing environment is strongly influencing the growth of the corals. And we were very interested in trying to find explanations for this. So that's what totally this question. This is also a question which is around for a long time. Some authors claim that the symmetry of a coral colony is controlled by genes. And some other people claim that it's entirely influenced by environmental influence. And an additional question is coming in this research, as I mentioned, so we noticed that if the flow is too low, if a coral comes into problem, that in the stream shape of the coral, where it cannot get rid of the surplus of the oxygen, then you get local bleaching. Okay, then the idea about this macroscopical growth model, which is influenced by the environment. So the idea is that if you take a blue coral, that has a lot of fur and which is very nice and visible, you can see, if you make a section to the colony, you can see the growth layers which are formed in different years. And then you can use it as a kind of like fingerprints, so it also gives important environmental information. And in this growth process, all the time layers are deposited on top of the previous layers. And the corallites, you can follow them as trajectories inside the coral. and they are slowly moving outward of the colony. And sometimes, if the colony in surface is extended, you can see that new corallites are inserted to get sufficient surface of the colony. You can see the growth of the many areas, particularly you see some factories, the species go out of the land. And from radiograph, you can reconstruct the original growth layers. And so in this growth process, in fact, the previous growth layers remain unchanged. We deposit all the time new layers of the previous layers. Then, so how is the surface of the coral looking? If, for example, if I go in the same species, not that still on the end, and then you can make the surface picture, you can see the arrangements of the coral lines. You can simplify it as a pattern of the polygons that you see over here. Now then we were very interested in kind of geometric representation of this growth process. We couple this with the influence of the physical environment. And then you should think about the diffusion, light, hydrodynamics, and how this is impacting the calcification process. And so this is the kind of concept of the idea that we have used. And so we deposit layers on top of the previous layers. The previous layers remain unchanged. And now the local thickness of a new growth layer is determined by the local absorption of nutrients or local light intensities along the surface normal factor. So if I deposit a new layer on top of the previous layer, and I construct first the surface normal, the new surface normal, and I try to calculate how much food is present or how much light is present, and it uses this difficult information to construct the length of the surface normal and to construct the thickness of this growth layer. So in the growth process, we all the time constructing layers of triangle. So we are going to represent the layer of the simulated coral. It's a layer of polygons, the subcomponent of the triangle. The previous layers remain unchanged. The triangles are again organized in polygons. The idea is that simply the polygon represents a simulated polygon. And now the thickness of the new area is determined by all kinds of environmental conditions. For all kinds of other things. But as an initial object, we use triangulated sphere. We also see that we use this in cell-based knowledge in a much lower level of abstraction. And in the growth process, if you start with a spherical object and you add a new layer on top of it, then you can, for example, increase or decrease the surface. And so what you have to do is you have to correct for this. And we simulate correlates and we assume that the polygons almost have an equal size. So if the surface is extending too much, you have to insert a new coral. And you can do this by using updating your mesh. Now, this is a little bit more technical slide, how we do this. So, if you have some mesh, then you can insert new triangles. Keep the triangle more or less the same time. And if you can also have cases where the surface in fact is decreased. And this is, for example, happening if the branch is formed, and then some of the triangles are becoming smaller and smaller, for example, at this point. And at certain points, they have an unrealistic size, and you can fuse them with each other. In fact, you delete a simulated polygon. But this is the whole idea of the simulation. So, when you have the three-dimensional simulation block, we have a substrate plane, we have a source plane for which we use simulated heating. You can have light in this box. The light direction. Okay, make this as simple as possible. correspond to perfectly. We can also introduce flow in this simulation box. That's the bit of this. You can see the flow of the alternative lead to left to right to right to left. And this is the basic idea that the diffusion, which we use the diffusion equation, which is complete concentrations. So we can solve the diffusion equation in 3D, and so we can assume that there is a source of Newton in the box. We can assume that at the surface of the simulated growth volume, that the nutrient concentration is zero, that all nutrients potentially are absorbed. And that means that we can also measure at every local point in the simulated object, we can measure the local absorption of the simulated nutrients. And we can use this information in determining the size of the new mean normal factor that we can construct to the surface of the previous growth area. And then, for example, in the diffusion limited conditions, the thing that we see is if we start with the we quickly get a branching object. And please note that there's no branching rule at all in this model. And so by in diffusion limited conditions, we get branching as a kind of emergent phenomenon. So there's no branching rule at all in this model. It's just an emergent phenomenon that we know. So over in the left picture, you see exactly a simulated object. It shows the initial starting point, the sphere, and it's exactly in 3D, and you see the growth layers which are constructed properly. And in B, you see the environment, the object, which are the core identification of nutrients. If you are close to the object, you need to identify some environment. And if you're at the top, that's the highest nutrient concentration. And this distribution is computed by solving this equation that you see over here. Over here you see the full branching object. Over here you see the section. So there's no branching at all in this model. So we get automatically branching objects in the diffusion limited conditions. So then because it's also known that they can transport nutrients over the surface. So if you have polyps at the surface, so it's also known that for example, sugars, which are produced by one polyp, they can be translocated in neighboring polyps. So also over the surface, over the colony, the kind of transport process is going on, in which nutrients are translocated from the neighboring polyp to other polyps. And you can describe this as a surface diffusion process. So now surface diffusion over the surface. And that can be modeled with this equation. And now the different morphologies which we can produce with this model. So over here, effectively the simplest step, you start with an initial material. You have diffusion limited growth. You immediately get a branching form. You can modify this a little bit with more bigger branches. And then what you can do is, and then you have to go in this direction. So from top to bottom, you can increase the surface diffusion. And then what you see in these models, if you increase the surface diffusion, and you see that increase the surface diffusion, you get more spherical objects. So the funny thing is that with a relatively simple model, with relatively simple efficiency, then you can produce a whole range of morphologies which you also can find in the development. And you can take this massive growth form, branching growth form. Now, we can also include light in we can make a simple three-dimensional light model, and you can compute the light intensities, sort of the surface. And you can also use this information for the velocity of the growth process. And so over here, you see the branching of it. In this case, you see a feasibility of the computed light intensities. You think which is very obvious because they want to be especially the tips will receive the highest amount of light. You can also compute shading because the upper branches are shading the lower branches. So, for example, in these lower branches, in fact, they don't receive any light anymore. But if the growth process is driven by local light intensities, you can predict a very slow growth process in the lower branches. And based on these observed light quantities, you can predict that especially the red parts of this branching plane will develop the highest growth. And now we mix everything together. We now assume that the growth velocity is a kind of mixture of sufficient light and sufficient observed nutrients. And then you can again use a whole range of different morphologies. And for example, some of the madrasa species which we are investigating, they seem to be approximated by this kind of shapes. Okay, then of course we have the big question. So we have simulated growth forms and we have real growth forms. So with the real growth forms, so we made a lot of computer tomography scans. And the nice thing about the computer tomography scans is that you can reconstruct the morphology of the coral in your computer. And we have the simulated morphologies. So until now, I was just saying, it looks a little bit like a coral, it looks a little bit like a different species. But we are also interested if we can really confirm this. But one of the first tests which we did was simply a visual comparison. So I put a number of simulated corals on the screen and a number of real corals. And then a question is for one time to coral biologists if they could answer the question, can you tell me which one on the screen is simulated and which one is real? And maybe this is a special question for you both who never have seen this slide before. Can any one of you guess which of the pictures in the screen is real and which one is simulated? And preferably also with some underlying argumentation, why you think so. Does any one of you have an idea? Any guess? Does any one of you have a suggestion? Which one is real, which not? Francesca, what do you think? Which one? This one? Yeah. What did you say about the right one?
I don't know how this works very well, but maybe we can have access to
Just If you just use your biological intuition, which one do you think? Yeah, which one do you think is real and which one is human? Any suggestion? This one? Is it real or simulated? Yeah? Which one? This one is all. And the cool one? They're all real. So I'm just cheating things, I'm not showing any simulation. That's all right. Okay. Okay, you both think so. And the other ones are real. Yeah, so it's very funny. So I asked this question to many coral biologists, and they always end up with a kind of similar conclusion. Can you also give arguments for why you think? Yeah. Yeah, yeah. That's exactly the argumentation which I've heard. Yeah. Yeah, okay. Really, really interesting. Yeah, very good argument. Yeah. So, so many of the coral biologists use the argument that the top one looks a little bit artificial to regular. The left one is a little bit more irregular, so it's probably a real organism. And about the other object, there is always discussion about. So, so the answer to the question is that the top one is a real coral. The two bottom ones only exist in the computer, so they're completely simulated. And just to show you that I'm not cheating, so here you see the real coral. So, and the only thing that we can do with the real coral is a reconstruction, where we can rotate it around. I would love to go look in the skeleton and find the growth layers, but it's one of the problems which it's never solved very well. So I can only show you the overall morphology. From the other object, you have the entire growth process, because you simulate the cell. And over here you see the entire simulated growth process. Just the conclusion that it's really a simulated. And so we also have been experimenting. So again, with parameter variation studies, what kind of different morphologies we can produce. And over here you see the kind of diagram in which you vary the influence of one intensity and and you get all kinds of different morphologies. So you can create simulated morphospaces. Here you see another example of this. And then, let's just check the time. Yeah, so I think it's maybe useful that I skip this topic about how to model the hydrodynamical flow. If anyone is interested, of course, I'm willing to tell more about this. But so I would now like to move in fact to the next question. And so over here, I'm making kind of a visual comparison between simulated growth forms and real growth forms. Of course, you cannot write a paper nowadays just by only showing a visual comparison, but that's not sufficient to be convincing. So you should also come up with a kind of quantitative approach, because you can say how much a certain growth form resembles another growth form. So that's another topic in which we have worked for a long time. Now I would like to move to that topic. So that's about the quantitative method, three-dimensional forms, and especially growth forms which have a complex branching shape. And how we can compare one growth form with another growth. Now, so if you look in the literature, so there are all kinds of different approaches that we can play with. So you have all kinds of methods based in global movement. For example, you might have seen papers in which people have computed fractal dimensions for these kind of objects. There are several other approaches. For example, a meaningful global measurement is the surface volume ratio of an object, which gives important biological information. The only disadvantage of global measurements is that it gives you lump morphology just in one number. And so it only gives you very limited information. So we are also very interested in local measurements. So if we could measure, for example, local curvature, measure the thickness of the branches, branching rates, and all kinds of related measurements. And we base these measurements on medical computer scans, and many of these scans with a colleague of mine in the medical department with Amsterdam. And for example, you see a collection of pictures of all kinds of different madrasa species. If you go to the Caribbean area, in the reach over there, you can find several madrasa species. Only a few experts are able to make a distinction between these four different species. But they have again this problem with morphological plasticity, so growth for changes with the environment. They have a very pretty and growth form, so it's a pretty complex shape. And for example, how do we distinguish this growth form in the right low only some experts are able to do it in the field. And on top of it, these balls have the computation that they can make rigorous. So they can visualize and make intermediate growth forms, which complicates things still more enormously. So we made a lot of selection, computed different species. Four of the species. There are even more madrasa species, but we focused on these four species. And then you have the DICOM image, so that's a special data format coming out of the computer tomography scan, which is usually used in medical scanners. And then you can do all kinds of things with it. So first of all, you can extract the surface covering the corner. But of course, you have also all kinds of information inside the platform, because you have a full three-dimensional representation. But then, how do you compare these things? So one of the methods which is frequently around, and I think it's still frequently used in biology, are so-called landmark-based methods. For example, we use comparison of different fishes, and we can put landmarks, for example, in open with spots, in the tail, and you can make these kind of pictures again from different pictures. And you can construct some kind of mathematical simulation. For corals, this is often work, and then take price. So these kind of landmark methods are very limited use for these kind of complex shaped organisms. So for this reason, investigating all kinds of different approaches. Then one of the basic methods which we are still using is basically this kind of idea. So we have the original corals, We put them in the computer tomography scanner. In the middle of the reconstructions in 3D in a computer. And after that, you can in fact simplify the volumetric representation of the object in so-called topological skeleton. And the idea is that we have this kind of lensing form, and we construct the so-called medial axis or the topological axis of the object. That is in fact a one-dimensional line, but it still preserves the topology of the object. And the nice thing is that you can use the morphological skeleton as a kind of roadmap to navigate to the goal and to connect it with all kinds of measurements. For example, I can go to thick, I can measure the local thickness of the branch. It's a very obvious thing. This is what you would like to measure in the core. Or you can measure the branching angle. The other nice thing about these three-dimensional reconstructions is that usually in biology you get with 4 or 5 measurements. And if you analyze the colony, then you end up with hundreds of measurements. So it gives you a very rich data set. So you can measure all kinds of properties. You can measure thickness. You can measure the angle between benches. And a very important biological measurement is the spacing between benches. This is also frequently measured in the field. It gives all kinds of important biological information. You can measure this automatically using this morphological skeleton. And then we have all this beautiful coral forms. And on top, we have converted all these beautiful forms into boring statistics, and you can do a kind of comparison. Then it's over here you see you have measured the branch spacing. And so over here on top you see the histogram. So you can see the frequency diagram of the different branching thickness. And one of these statements we immediately see in this distribution is that the compact shape has a very peaked distribution. One of the open object has an initial flattened out distribution. Now, this is all something that you can quantify in statistics and test. So we can use this as a kind of fingerprint of these kind of morphologies. And then so we were also very interested that we could use this approach for preparing forms. So over here we analyzed some of our simulations. And we now kind of parameter the thickness of the branch spectrum. And every point in the morpho diagram now corresponds to a certain morphology. And then the next step which we made is that we were also very interested how well can we approximate the different biomineralization species. And so a few slides ago I made this kind of visual comparison between mineralization realities and other simulations. Now what I'm going to use now in the branch technique for the y-axis of the branch spacing. And the color, we can do 4 different species. So, the blue is the advanced species. In purple, the Madras for moment, red is Madras for practice. In green, it's a special in the species in the green, Madras for learning. And the red levels represent our simulations. So what you can see is that our simulation mix nicely up with the Madras morphology. You can also approximate some of the Madras with Jacobus morphology. So, for example, you can see in this diagram that these guys are coming pretty close to each other. As you can see that the green ones we cannot capture So this is an indication that we use something in our simulation model to approximate this kind of morphology. And this is, of course, a very interesting point. Yeah, that's causing this difference. We also worked on a genetic comparison. These migration species. So then part of DNA from the nucleus and part of DNA from the mitochondria, from different samples as compared with each other. And this produced this extremely complicated morphology. So what you see is a very confused biology, 4 different species. So, for example, you can see that there is a picture with blue ones, but you can see there is also another species, the green one, and they're not in this picture. You can see that it's again a cluster of green one, which is again mixing up with the blue one. And now the explanation for this complicated process is hybridization. So, for example, this individual which is mixing up the green one that is mixing up with the blue cluster. It could easily be a hybrid between both species. And this is also something which we also could reproduce in similar kind of cluster diagrams based on the morphology. So we also could detect hybrids which only somewhere between clusters of different species. But then some recent work which we did is so another morphological property that is frequently coming back in all kinds of biological methods is the curvature of the shape. It's a 3-dimensional curvature, which can have all kinds of different values. But for example, here you have a point of high curvature, and over here you have flat surfaces. At this point, you have a negative curvature. The curvatures seem to be very characteristic for all kinds of biological objects. And we also developed a method, which was entirely based on these kind of curvatures. And we tested them out for a few corals, which are extremely challenging to recognize in field. I believe so. It's about these 2 Acropora species. I believe there's only one woman in the world who knows all these Acropora species. You can recognize them. I think for most people in the world, it's a very complicated process. So the question which we have, can we make scans of these objects again? And can we do the same frequently? So can we have before we make a 3-dimensional model of the scan of the body, and we reconstruct the skeleton. What's important to note that these scans were not made with the medical scanner, with the photographic scanner, but they were made with a visible light scanner, which is much easier to do. And so it's actually equivalent that we use the table, we use the camera, and we let the table below that very small angles, and all time pictures. And then you have a special software gluing together all these pictures and makes the 3-dimensional picture. And it's very effective, and that's really surprised by the good results. So our colleague, she did this for this period. And it's by the way, much cheaper also, of course. And you can even have handheld devices, which you can bring with you in the suitcase. So you can easily go to a different museum or maybe a different test site. You can use experiment. So these are all made with visible lights. The only disadvantage, which I should mention, is that we don't have any information inside the paper. It's only surface reconstruction. If you are interested in recognizing the different species, or if you are interested, for example, in following the growth process of the paper, this can be a very effective model. In theory, maybe not the process work for EEG, which we also have handheld laser scanners that you can use in water. You can, at least the theory, you could go over make recordings of the morphology at a certain point in time, come back within the year to make the same scan again, and make a comparison between the morphology. This could be a useful tool to monitor, for example, the health of a reef. But then about the curvature, so you can define curvature in this way. For example, you can define curvature later on in this way. And if the curvature is positive, like in the complex point, and if it's negative, you can use the same definitions in 3D, then you can recognize curvatures, maximum curvature, minimum curvature, and mean curvature. And you can also include the second curvature. So it gives you all kinds of things. If you measure the curvature, it gives you all kinds of possible measurements. Now then, we did this measurement of curvature in detail. So here you see, you see the reconstruction, as we call it, and you see the values of the different curvature. And you can see that, especially at the tips, that you would expect, and you will use higher curvature. So now again, we can collect a lot of measurements. And then you can compare three different species. So over here you see the species in the tree. Of course, there is suffering in this kind of morphological plasticity. And for example, we have species A and B. And so we know some core biology to distinguish in the tree. And then over here you see the morphology of the species in more detail. And so again, over here, we converted about using linear analysis, we were able to make a distinction between different species A and B. Of course, you can see it in the charts, and it's a kind of principal component of the analysis that you see over here. But you can see that there are still some points which are overlapping, which are really problematic. And with most of the cases, we can recognize with this analysis. Okay, so just a few very quickly. I've shown this classification model. I have also shown I think, the impressive list of hypotheses about classification. I think in the general conclusion, we think that we don't understand that much about it yet, especially about the underlying genetic regulation and the underlying physiology. So, in fact, most of the details about gene regulation, classification, are missing. So there is, of course, genomic information available, now calls, siblings, but so we will be very interested if it is reconstruct the regulatory network for the classification. So I've introduced some gene regulatory network models. You can also use didn't show this, but we can also use this method to investigate the real gene expression data and do a kind of reverse engineering and try to infer gene regulatory networks with this approach. I think an important message, I think, is that these kind of computational models are a very important way to organize data and scientific observations themselves. So by making, for example, this classification physiology model, we were forced to look at all the available hypotheses and put them next to each other and compare the different data. And already this process, even if the model finally doesn't work, I think is already very valuable. And if it works, so it's also a very good way to find the missing points of information. I think it's pretty obvious that it's still missing. And it also could provide a kind of guideline how you can collect data in a more systematic way. Currently, I think genetic information is collected. Sometimes I get the impression in a massive and almost haphazard long way. It would be very nice if you could especially collect, for example, gene expression data around things which you still don't understand, and which are very important. Okay, then I would like to enter the management. So over here on top, I have some of the PhD students who have contributed in this project. The list is not entirely complete, but for example, Amir Abdul did a lot of work on the reconstruction of gene regulatory networks in nanocrystalline potential. Carlos developed the first three-dimensional version of a cell-based model of nanocrystalline. And Helena Willard is currently recently started in my group as a PhD student on models of the classification picture. And Roland is developing this three-dimensional version of the cell-based model, and hopefully you will publish it very soon. And Paula, she did amazing work about, also together with Frederick, to isolate the organic matrix in a core. And in the middle row, you see some of my collaborators. And you see Rens Romans from the University of Cambridge. My collaborator Adrianus, collaborating in the development of biology for the nanocrystalline. And a picture of Jean-François Pro in the University of Brussels. And with him, I'm also collaborating in the course. And at the bottom row, you see some of the students who also played a very, master students who played a very important role in this whole process. And for example, Aizan is currently finalizing a master thesis about his model of the second derivative axis in the nanocrystalline. And Martin, he developed this regression model, a cell-based regression model for Kaandorp. And both Koens also contributed to this nanocrystalline regulatory network model. Okay, thank you for your attention. Any questions? Yeah. This? I know there exists a lot of work about this, so it should be around. Yeah. But we didn't try to evolve this genetic information in this case. So in fact, Catalina did so. She just used abusing the material. Yeah.
What's the piece in the like the communication
So I know that there are many papers around people, so they took a call in the fields, they measured the transcriptomics, and then they exposed the call to all kind of stresses, so like the acidification, and then they measured the transcriptomics again. And so maybe I don't understand this paper very well, but it gives a very messy and blurry impression. So all kinds of things are upregulated and downregulated, and it's very difficult to find an explanation for it. Also, you cannot follow the process really in time. Most of the cases, you have long observation compared to an actual other observation. And if you do this for so many genes, what do you know? So I think these kind of things make much more sense. For example, if you could do observations with PCR and follow a few important genes in time. Special.
Just starting this complex is indicated.
Yeah.
We wonder why the biological complex is so complex. Because the advantage is complex.
What's the value?
The advantage of complexity, why the complex must be so complex.
Yeah, so my explanation for that, for example, the gene regulatory network, we call it complicated. So it's maybe an important distinction between things which are complicated and complex. So complex is also used many times in mathematics, the complex systems. Usually they're constructed in a very simple way, and they produce all kinds of strange behavior, complex behavior. And I think the gene regulatory networks in nematostella, I think they have unexpected complications. But my guess is that it has to do with the unbelievable robustness of the system. It needs so many gene regulatory networks to be absolutely sure that nothing is going wrong. You can regenerate and repair everything.
It's also the known.
Yeah. There's probably a lot of redundancy, but you don't know, right? So maybe part of your regulatory network, which is now redundant, it may be super useful in the different conditions. Does this give an answer? And I think the other thing is that what's always remarkable is that from very simple systems, we can also produce very complex shapes with growth forms. And these microscopic growth methods, they are not very complicated. In fact, they're just based on a few years. But we can produce a lot of complex shapes. That's a little bit the other side.
This is such a But it's still, what? It's still like, what is going to ask other individuals?
Yeah.
So what about girls?
Yeah, so the strange thing is that so you even see it in different librarians that they follow different pathways. But I also have stories about Adrianus. I think they're very related species, and sometimes they take very different regulatory pathways to the destination. So then, for some reason, some related organisms were able to change this in a very fundamental way. Which is totally and maybe these things also have to do with the way of living. So if you are attached to the substrate, then you have a different way of living. You try to move around. So, for example, you have Drosophila. These Drosophilans, they have a kind of direct pattern of development. And it makes sense if you live in a bin and rotten fruits, you have very little time to develop new things very quickly. Any more questions? Okay. Thank you.
Accountable future.